Now I'm talking about the reconstitution of the molecule test motilities. So then the molecule test is the class molecule test. It includes mycoplasma and the genus. So then the first author is the Hanakiyama. She is the PhD student of our lab. And this is the collaboration with Shige Kakizawa at the ICE Japan. And I'm Makoto Miyata from Osaka City University. And this study was also helped by our lab members. Here is the cell evolution focusing on motility. So then this was suggested by us in 2019. And this is the origin of the cell. And so they acquired the peptide glycan layer and also the DNA segregation. So then they became the ancestral bacteria. And we don't know the relationship between the bacteria and the archaea, but archaea lack of the peptide glycan layer. So then they have the flexible peripheral structure. So then they acquired membrane remodeling. So then they became eukarya, as you know. And a small group of the bacteria, as you know, so they quitted the peptide glycan layer to avoid, to evade the host natural immune system. So then they became class Mollicutes. And at the same time that they need to move for the survival, so they achieved the new types of the motility systems because usually the bacterial motility systems depend on the peptide glycan layer. We focus on the synthetic cell, the SYN3. So then we are trying to reconstitute the motility systems of the class Mollicutes. So then it will be done by our group, and another, Professor Shiomi is trying that peptide glycan layer, and another group that Professor Robinson is trying reconstitution of the membrane. It means so then the reconstitution of the cytoskeleton system. So then our project is supported by the CREST project by the Japanese government. Today, I focus on the project of our group, the reconstitution of the class Mollicutes motility systems. This is the real-time movie of the spiroplasma swimming. Spiroplasma belongs to the class Mollicutes. This looks like the spirochetes, but actually this mechanism is unrelated to the spirochetes swimming. And here is the schematic. So here is the image of the video. So then here is the schematic. Basically, they are in left-handed helices. So then from the front side, they shift from left-handed to the right-handed. So then the shift the travels along the cell axis. So then again, from the front side, it shifts from the right-handed to the left-handed. So then you think about the helicity shift, the back side of the shift should rotate. So then in this mechanism, these bacteria, pushes the water to the backside and they swim forward. We focus on 7 candidate genes involved in this swimming system. So then the first one is fibril, and the other 5 genes are MREB. So then you know that the MREB belongs to the actin family, and here is another gene inserted inside of the MREB, the cluster. And we intended to the reconstitution of the spiroplasma swimming by using the SYN3, so that we assemble these genes into the genome. So then we see what happens. Now here is the typical video showing that the reconstitution of the swimming. So the leftmost one is the spiral plasma swimming. It's a kind of goal. The second one is just the video of the scene 3B, so it doesn't move by itself. And the rightmost one is the swimming of the swimming scene 3B. You can see the swimming was reconstituted in the scene 3. And so then we analyze the cell morphology. So then here is the schematic of the spiral plasma cell. So then you can see that, that they share, I'm saying the spiral plasma and the swimming sensory, they share the width of the helix and also the pitch of the helix. So then we examine the structure of the cells. So these are the typical image of the cryo-EM. You can see the cell membrane like that. So then, and also it's a little bit difficult to see, but we can see clearly the filamental structure at the strong curvature parts for the both cells. And also we analyze helicity shift. Here are the half speed of the video of the swimming. So then we picked the frames and straightened the cell by the computer. So then we align these images like that. So then we can find that the helicity shift travels along the cell axis, and the traveling speed is for the spiral plasma cells. It was about 9 micrometer, and also the rotation speed is 12 rotations per second. So the number of the swimming the sensory was similar to the spiral plasma cells, but something is different. So then we assign the left-handed and right-handed part. So then we found in the spiral plasma, so then the shift is more continuous, but the swimming sensory, it's a kind of the discontinuous. So then it's reflected to the swimming behaviors. This is the trace of the 10-second of trace of the spiral plasma swimming. And then you can see the spiral plasma cells can travel to the distant place. But the swimming cells, swimming the sensory, they don't travel to the distant place. And this should be caused by the cell structure. So here is the typical image of the spiral plasma cell. So this is sensory. Then you can find easily that one side, one pole of the spiral plasma is the tapered, as indicated by the red triangles. So then we try the silencing of individual genes by introducing the nonsense mutations. And here are the same thing with the video I showed previously, the spiral plasma swimming and the swimming the sensory, and this is the original sensory. So then, interestingly, the silencing of the individual genes didn't affect so much for either of the helicity or the swimming, but only MREB5. So then they showed the obvious effect on the cell shape, and also that they never the same anymore. So then now that we are the detailed analysis are ongoing. So then I'll introduce another subject which we are now starting. So as you know, so that we have been working on mycoplasma gliding motility. So this is the mycoplasma mobile and the mycoplasma pneumoniae. So they just I just introduced what is mycoplasma gliding to the people who are not mycoplasmologists. So this is probably I showed this video someplace sometime to you, mycoplasma people. But anyway, so this is mycoplasma mobile gliding. Then they glide. This is the real-time video. So then we put Triton. So then Triton damages the cell membrane immediately. It stops the cells keeping on the cells on the grass. So then we remove the nucleic acids because they inhibit the gliding, working as strings. Then we put ATP. In just 5 seconds, they started the normal gliding the motility with normal speed. So we are now trying this one. Yes, that is. Thank you for your attention. So then I'm happy to do any discussion with you. Thank you very much.
Makoto, this is stunning. I would never have guessed this would have worked. I don't even know what to say. Someone help me. This is just dazzling work. Questions, anyone?
No questions?
I mean, did you expect this to work?
Yeah, it's a good question. Yes. So then actually we tried the expression of the individual proteins. So then one day but I didn't have enough time to show that. But some of the proteins change the cell structures efficiently. So then we decided to try to the input express all of the candidate proteins. So then it worked. We are very happy about that.
I have a question for Makoto. If
yes, go ahead.
Kim here. Hi, how's it going?
Nice to meet you.
Yes. Great to see you. Do you have any idea how this new activity you've put in, how does it affect division of the cell? Because I presume I'm not exactly remembering how spiroplasma divides, but is there any influence on that whole process?
Yeah. Actually, we chose the good-looking cells. So then, when we grow the spiroplasmas, so then they show the very uniform the cell length and so the cell shapes. So then, in case of the swimming the SYN3, so then actually the cell length is not so much uniform, and some cells that can form very easily the cramps. So then it's a kind of we need some technique to disperse the cell, and so then optimize the condition to observe the swimming.
Yeah, given the fact that 3.0 has a very bizarre, as James I think Pelletier showed.
Yeah, probably we need some the proteins to reconstitute the orders the cell division, and also as I told you, so then we don't express the the proteins involved in that the tip. I'm saying that we believe that the tip proteins decide the polarity of the cell. So then we don't know, we have no idea about the proteins responsible for the polarity.
Yeah.
Yes.
Fascinating.
Yeah, thank you.
It would be interesting to see if you could get an interactome of the swimming SYN3B and compare it to our colleagues at, so a couple of talks from now, we're going to hear about the minimal cell interactome, but I would wonder what proteins, of the spiroplasma proteins you added, what they might be interacting with in SYN3B, or if it is just a completely independent mechanism.
Yeah, so that's good question. So then we believe that only these proteins are enough to reproduce reconstruct the swimming motility. But actually, we don't we have no information. Is there any other SYN3 the proteins that helping the swimming? So then it would be the future study. Yes, I agree with you.
Because spiroplasmas and the mycoides group are not that
it's a kind of the close. So then we should be careful about that.
Thank you very much, Makoto.
Yes, thank you very much. Thank you.
Dazzling work. I'm just,
Thank you.
I am stunned at what you showed.
Yes, thank you very much.